everyone, my name is Hasminam Sampilo and we're gonna tackle about chromosome. So first, what is chromosome? Chromosome is a string of DNA wrapped around associated proteins that give the connected nucleic acid bases a structure. During interphase of the cell cycle, the chromosome exists in a loose structure, so proteins can be translated from DNA and the DNA can be replicated. During mitosis and meiosis, the chromosome becomes condensed to be organized and separated. The substance consisting of all the chromosomes in a cell and all their associated proteins is known as chromatid. In prokaryotes, there is usually only a single chromosome which exists in a ring-like or linear shape. The chromatin of most eukaryotic organisms consists of multiple chromosomes as described later in the article. Each chromosome carries part of the genetic code necessary to produce an organism. Having the entire genetic code divided into different chromosomes allows the possibility of variation through the different combinations of chromosomes with different alleles or genetic variations that they contain. The recombination and mutation of chromosomes can occur during mitosis, meiosis, or during interphase. The end result is organisms that function and behave in different ways. This variation allows population to evolve over time in response to changing environments.
Hi everyone, I am Zafir and Mustafa and we will be talking about the function of chromosomes. Chromosomes facilitate proper cell division and replication. The main function of the chromosome is to fit the DNA inside the nucleus. As we all know that our DNA is too long. If we unwind all the DNA of a cell, it is up to 2 meters in length. Hence, it is very important to fit it inside the nucleus, which is facilitated by chromosomes. By interacting with proteins, DNA forms a coiled structure chromosome. Chromosomes also help in inheriting genes or DNA from parent to their offsprings. Furthermore, sex chromosomes decide the sex of the embryo we had discussed it above. The process of sex determination and sex differentiation is governed by genes located on autosomes and sex chromosomes.
Size and number of genes on different human chromosomes. Chromosome, number of genes.
A total of around 21,000 to 22,000 genes are present in the human genome, in which chromosome has the highest number of genes, which is 2,000, while the Y chromosome has the lowest number of genes, which is 200.
My name is Altamira S. Adam from 9STA and I'm here to discuss about chromosome structures. I'm going to discuss about chromosome structure. You're probably wondering, what is chromosome structure? Well, as seen in the graphic above, chromosomes have a very complex structure. DNA, or deoxyribonucleic acid, makes the base of the structure as seen on the far left. DNA is made of 2 strings of nucleic acid base pairs. The base pairs in DNA are cytosine, adenine, thymine, and guanine. The spiral structure formed by the 2 strings of DNA is due to complementary pairing between every base with its pair on the opposite string. Adenine pairs with thymine and guanine pairs with cytosine. The opposite side of the bases form a phosphate deoxyribose backbone, which keeps the strands intact. It is duplicated, the strands are separated, and the polymerase molecule builds a new string that corresponds to each side. In this way, the DNA is perfectly replicated. This can be done artificially by a polymerase chain reaction in which special enzymes and heat are used to separate and replicate the string a number of times to produce many copies of the same DNA. This makes it much easier to study any string of DNA, even whole chromosomes or genomes. After the cell has expressed and duplicated the DNA, cell division can occur. This occurs in both prokaryotes and eukaryotes, but only eukaryotes condense their DNA so simple that relatively few structural proteins are associated with the chromosome. In eukaryotes, many structural proteins are used. The first of these proteins are core histones. Many individual histone proteins bind together to form a core histone. The DNA can wrap around one of these histones, giving it a wound structure. This structure and the associated histone is known as the nucleosome. Seen in the third picture from the left, this nucleosome form leads on a string. The string becomes wound back and forth by another histone H1, and eventually fibers are produced. The next type of protein, scaffold proteins, start to bind the fiber into a loose structure. When the chromosome must condense during cell division, more scaffold proteins are activated and the structure becomes much denser. In fact, even with a microscope, individual chromosomes cannot be discerned until near the middle of cell division cycles. When the chromosomes become very dense, this process is seen as the picture's progress towards the right.
Examples of chromosome.
Good morning, I'm Eukaryote Replication from 9STA. The first example is Prokaryote Replication. When a single bacterial cell has reached a large enough size, it can reproduce asexually. Although there are no membranes to separate individual organelles in bacteria, the cell will duplicate its DNA and special chemical it needs to survive. The DNA exists in a single chromosome, sometimes called genophore, which is replicated by the individual strands being separated by polymerase building new corresponding strands. The two chromosomes are separated into individual cells, and the cells carry out their function by creating proteins from the DNA and interacting with the environment.
Eukaryote replication. A much more complicated view of chromosomes is present in eukaryotes. In eukaryotes, the DNA is replicated at the end of interphase, the part of a cell cycle in which cell grows and functions. Much like in prokaryotes, the DNA strands are separated and the new strands are created by enzymes. However, in eukaryotes, there are multiple chromosomes. The new chromosomes remain connected at the centromere, a structure that allows microtubules to connect and holds chromosomes together. These are now known as sister chromatids because they are identical copies. These chromatids can undergo some variation during meiotic cell division when recombination can occur.
I still remember loving the danger. You set me on fire.